So in few days, 4 days, 5 days, they will move to the next layer. But in few cases, exceptional cases, such as a virus, you have an enzyme which allows them to move across the membrane. So let us go back and see how we can test that this membrane protein or membrane lipid, they are fluidic in nature. To understand this, a very simple experiment called as a fluorescence recovery after photobleaching technique, that is the FRAP technique, which was used. And in this technique, what is done is that here lipids and many membrane protein are demonstrated that they are moving across the membrane. Just to show it graphically, I have taken this picture, and here suppose this is a cell. In this cell, this is the membrane which you are looking at. Now what you have to do in the first set is that you label the membrane. So you take a fluorescent dye and label it. So your whole membrane is labeled. How does it look like? It will look like this. So when you see under the microscope from the top, you will see that whole cell outer surface is having a fluorescent dye. And if you look at the cell, it is the outer layer of the cell, that is a membrane which has been labeled. Now in the next set, what you do is that you take this structure and focus a very small area, and in this area you pass the laser beam. And when you pass the laser beam, what you expect is that the protein in that area are degraded. And if the protein are degraded, this becomes a bleach. Now it is bleached. What is the next? Do nothing, leave it for a certain period of time. And during this time, you have to just maintain that temperature is something like 37 degree or normal temperature of the room. Under those conditions, what is expected? Because as you know that the membranes are fluid, what will happen? The fluorescent dye, they will move from their present position to everywhere. It is not necessary that they will move here. So during the process of movement, that bleached area will be again become fluorescent. So this is the way you can monitor this. And to show this, the same experiment has been shown here, and to show what you actually going to measure. So this is a cell, you have bleached it. So this area you have concentrated, bleached it, and after a period of time, this again recovered. Now, if I do experiment, what I have to see? I have to do and check the fluorescent intensity. So this fluorescence intensity here, the one which is the flat line, shows the 100%. So whatever the fluorescence you get, you consider this as 100% fluorescence. And at a given time, indicated time, you pass the laser. So during the passing of the laser, what has happened? The protein has got degraded, and because of the degradation of the protein, the fluorescence level will come down. So you have fluorescence level here. Now you did nothing and you left it, and then the recovery process will occur. And then after a few minutes or after an hour, depending on the movement of the protein, this will come back to the normal. Now consider a scenario where membrane lipids or membrane proteins are not fluid. And if they are not fluid, what you expect is that once you have bleached it, there should not be any recovery, isn't it? So if you bleach it, if the membrane proteins are not moving, which was labeled earlier, so that will remain bleached because other proteins, they are rigid. So they will remain at that place and this area will remain bleached. So since they have moved, that is why the bleach has disappeared and the color has reappeared. So that you monitored by the fluorescence. Now, by doing this, can we quantify? Answer is yes. So what you have done is that you have looked at the lateral movement of the membrane protein, and it has been visualized by fluorescence microscopy using a technique which is a FRAP. Now, the rate of recovery of fluorescence depends on the lateral mobility of fluorescence labeled component. So it is a protein or it is a lipid, whatever you have labeled, depending on that, how fast they're moving, you get a diffusion coefficient. How it is calculated? S is given here is the average distance which has been traversed in time. So S gives you the time uh, in a particular time how much distance has moved. So this distance you can calculate if you know the value of a d and the time. So it is a root of 4 dt, and that is what comes as the average distance. So you can find out that, for example, in 1 second, how much labeled compound could be lipid, could be your protein has moved. Now, the question here is, what is your expectation? Lipid should move fast or protein should move fast? And why? Any one of you? This Can you hear me?
Yes, sir, we can hear you.
Okay. So my question is that if in a membrane I check whether lipid is moving fast or protein is moving fast, can you tell me which one should move fast and why? So suppose you have a viscous solution, and in a viscous solution, so membrane protein is nothing but, membrane protein as well as lipid complex is nothing but a viscous solution. That's what you expect, that it is a fluid. And in this fluid, what I'm asking is that protein is moving fast or lipid is. So you have a protein, it is not shown here, but embedded protein you will be having, whereas these are the lipid. These are, lipid molecule, phospholipid mostly.
So is it lipids?
Lipid. Why lipids?
So just a guess.
Okay. It could be a right guess, but the answer is that lipid, being a smaller molecule, they will move faster. So anything which is small, they will move faster compared to the proteins. Why? Because protein, if you have high molecular weight, their movement, they will be restricted, whereas the lipids, they can move faster. In fact, one can determine and track the time and diffusion rate of lipid as well as a protein, you will be finding it's quite high. Let us guess. Anyway, it is written over here. So suppose you have a lipid which is present over here at any point here. What time you expect that the same lipid should come back after moving to this place? Suppose I have a cell of E. coli. Normally, it takes a second. So you can imagine in a E. coli bacteria, the movement of the lipid is so fast that within a second, it can, move across the cell and come back to the same position. So this is what gives you some idea. And it depends on the diffusion coefficient. So for each protein or lipid, you can calculate the diffusion coefficient. And based on that, one can calculate at what time they will be, coming back to the same position. Now, the question is, does this membrane remains always fluid, or can we change the fluidity of the membrane? And second question is, what is the component in the membrane lipid which is maintaining the fluidity? As you know, the fluidity is a major, the thing which is going to maintain the fluidity is basically your lipid portion of the membrane, not the protein. Protein are the one which are working, they are the one which are actors, and the lipids, they are the one which provides the environment where this action can happen. So to answer this, uh, we have, of course, this part we have already discussed, that the movement occurs in the lateral one, not the across the membrane. Now, another point which needs to be discussed here is that why such kind of a restriction occurs. Why they are not moving. Suppose there is a lipid, say there is a phospholipid. I said they move laterally, they don't move from this place to this place. That means transverse diffusion is not possible. Answer is a very simple, because if I have this molecule to move across the membrane, what you have, they have to come through the fatty acid chain of other lipids. And this portion, as you know, is a hydrophilic. So now what you are expecting is that a hydrophilic molecule should move across the membrane through the hydrophobic groups or hydrophobic fatty acid chain and then reach at another position and come back to the same structure. Answer is here the cost of energy is quite high because the head group is hydrophilic, and that's why the movement of lipid is restricted. Now, if I talk about the proteins, in the proteins, suppose this is an integral protein, what happens that though the part of it which is embedded in the membrane, they are hydrophobic, but the part which is outside the membrane, they are hydrophilic in nature. And if it has to rotate or it has to, cross the membrane, in that case, what will happen? Though the rotation is a possible, in some cases, rotation is a possible, but in most of the cases, this hydrophilic group, when they cross the membrane, the cost of energy is very high. And that is why they are not moving from one direction to another direction unless there is something which lowers the activation energy barrier. So it's actually activation energy barrier which does not allow the phospholipids, their head group, to move across the membrane. And that is why it does not allow the proteins which are present in the membrane to move across. So that is what you see, that flip-flop of the protein molecule has not been observed. And if it is not observed, what will happen? If it is not observed, there is asymmetry which will remain. Asymmetry means that the lipids which are present in upper half, they will remain in the upper half. The lipid which is present in the lower half, they will remain in the lower half. And same way, the protein which is present in the upper half of the membrane, they will remain in the upper half. The protein which is present in the lower half, they will remain in the lower half. So this way, they maintain their asymmetry. And suppose there is a flip-flop movement, as has been observed as a rare example in case of viruses. What will happen? Whatever is present in the upper half, they will move to the lower half, and after a period of time, suppose X amount of this lipid is present here, they will, become equilibrated with the same amount of the lipid over here. So asymmetry will be lost. So that is what is not happening. Under a normal condition, the lipid, they remain asymmetry, the protein remain asymmetry. And when I say asymmetry, that means they will the composition of a membrane in the outer part and the inner part are different, and they remain whatever were in the pre-existing membrane. Now, coming to one more interesting point about the fluidity of the
Sir, I
membrane.
have a doubt.
Yeah, yes.
So in viruses, the flip-flop motion is allowed. Sir, how does it benefit their structure, membrane
Uh,
structure?
I cannot give you the real example, but yes, they have additional set of enzymes which are known as a Philippase enzyme. And these Philippase enzyme, they allow them to move from one direction to another direction. In general, if you look at the viruses, viruses are supposed to be not the one which are living in its own machinery. So their machinery is dependent on where they are invading. So I do not know actually whether it is the advantage for viruses or this is happening where Philippase has a different role as well. But yes, the presence of enzyme that changes the composition of the membrane in viruses, and this kind of a change is not present in other organism like bacteria or in the animal cell. And this kind of maintaining the asymmetry maintains the job of the membrane. In fact, if you look at the viruses, they don't have any job to do. They do not have any machinery. They don't have a machinery for example, the energy synthesis. They don't have a machinery for uptake of the things. What they do is that they integrate in the uh, in certain membrane, whether it is a plant membrane or plant cell membrane or animal cell membrane or bacterial membrane, and then they give its genetic material inside the cell, and then genetic material they synthesize everything. Now, from where they synthesize, the machinery is coming or energy is coming from the host organism. So that is why the viruses, they can afford to have the change in the system. Now think of a scenario where in animal cell this kind of flip-flop movement occurs, and the the protein which is required inside the mitochondria, they come out on the outer surface or outer membrane of the mitochondria. So energy will be synthesized outside the mitochondria, and there will be waste of energy. You will learning there is a very tight regulation of energy, how they are synthesized, how they are kept inside the mitochondria, how the flow of energy, that means ATP, occurs from inside mitochondria to outside mitochondria. So that one function I talked about. There are many functions. So that is why I guess that the bacteria as well as animal cell, they maintain the asymmetry of the membrane. And that also maintains that initially, say, parental membrane has asymmetry, the same asymmetry is maintained in the daughter cells. And this kind of asymmetry also regulates, suppose I have the bacterial cell or animal cell. So suppose in the outer part of the membrane, there are some transporter in a particular structure. They are supposed to bind with the ligand, and that's the way they take the energy, and not only energy, they take the material which can produce the energy. Say transport of carbohydrate, transport of amino acid, transport of ions. All those are dependent on the membrane asymmetry, because the same thing is not present on the inner surface. Am I clear?
Yes, sir.
Okay, so let us move on to membrane fluidity. How it is controlled? Normally, the fluidity which we are talking about can be controlled by the fatty acid and cholesterol. So this is the statement which I am giving. Now you tell me, other than fatty acid or cholesterol, if I want to maintain the fluidity of the membrane, what else we can do? Suppose you have oil in your kitchen and it is flowing, and you want to stop the flow of the oil. What can you do? You cannot change the composition immediately. You cannot add something. What else you can do?
So we can regulate the temperature if we are allowed to regulate the external conditions.
Yeah, that is the best thing. Suppose you have a coconut oil. You see in winter it becomes solidified, and in summer it becomes fluid. So very easy thing is that you can maintain the temperature. So I'll be talking reverse about this a little bit. And other than that, the inside the cell, you cannot change the temperature. For example, inside our body, I cannot change the temperature abruptly, even to 2 degree, 3 degree temperature. That creates, fever. So the maintenance of the fluidity inside the organism can be maintained by the fatty acid and cholesterol, which are component. And we have already discussed, for example, here on the left hand side, you see that these are the fatty acid, and this is the fatty acid which is 18 is to 0. It is called as stearic acid or stearate. And this is the one, same one, but having one unsaturated bond. So you have 18 is to 1. That is called as oleic acid. So these are two different fatty acid having same number of carbon atoms. That means chain is same, but they differ by one double bond. Now what has happened by presence of one double bond, that the packing of this fatty acid is poor, whereas the packing of this fatty acid is very good. On the other hand, I can take an example where instead of 18 carbon chain, I take a carbon chain which is only 6 carbon atom or 8 carbon atom. So in that case, the packing will be less dominant than this one, because here the number of carbon is large. So there are two things which are maintaining the packing. One is the number of carbon in a fatty acid, and second one is the presence of a double bond. You can have a two double bond, you can have a three double bond. So you can have 18 2, 18 3. So if you increase the number of a double bond, their packing is less compact. If you increase the chain length, packing becomes much more stronger. So by these two manipulation inside the body can maintain the Do you require the membrane to be too fluid or too solid? Answer is no. What you require them to have a moderate fluidity where protein can move, but at the same time, they should not become so fluid that these proteins should leak out. Suppose I make it too fluid, what will happen? The protein, they will not remain. So you can imagine the fluidity of olive oil and water. So one is slightly restricted, the movement, another is a free flowing. So, or alternatively, you can imagine the coconut oil at, say, temperature where it is not completely solidified nor it is completely melted. So you see a kind of a change in the viscosity or a change in their fluidity. Now coming to the point that which we are talking about, suppose I do this experiment. Let's go back and try to explain the phenomenon a little more. I do this experiment, and when I'm looking at the recovery, so I have bleached the cell as you have done in the previous experiment, and here when the recovery is occurring or when I'm leaving it after bleaching, I freeze these cells. I make it at 4 degree or make it at 6 degree. What you will find is that the recovery does not occur. Why it is so? Because membranes, they are not fluid. So if you want the recovery, you have to keep it at a temperature where the membrane lipid is making it fluid. So at what temperature you think they should be fluid, or they will be moderately fluid? The temperature at which the organism grows. So suppose your E. coli is growing at 37 degree or 30 degree, that is the optimal temperature for the fluidity of membrane. Now, though I have not written the question, let me just pose a question and try to explain it to you. So here, this thing is given where it shows that red one shows the fluid-like structure and the blue one shows the solid-like structure. So there is a temperature, and this temperature is known as a melting temperature. And here, what you find is that the transition from rigid to fluid state occurs rather abruptly as the temperature is raised above Tm. So above this Tm, what you find is that there is an abrupt change in the membrane fluidity. The transition temperature depends on the length of the chain, as I said, number of double bond and length of their fatty acid chain. In animals, cholesterol is a key regulator. I'll talk about how cholesterol does it, but what cholesterol does is that it helps the maintenance of the fluidity. Now, I'm giving you an experiment and let us try to discuss that out. Suppose you are growing your E. coli at a particular temperature. Say you are growing it at 30-degree temperature. Now they are happily growing and you know that they have a proper fluidity. Now I have done an experiment separately where I have grown at 30 degree and at the 4 degree, or maybe 4 degrees too low. Let us put it at uh, 10 degree. So one is at 30 degree temperature, another is at 10 degree temperature. There is a sharp gap of something like 20-degree temperature. Now I have done a clever experiment here. What I did is that because the bacteria requires carbon as a source for their growth, what I did is that in both the medium, I have put the fatty acid as a sole source of energy. Now here, I have put a combination of fatty acid. I have put short chain fatty acid, long chain fatty acid. I have also put the fatty acid containing one double bond, fatty acid containing two double bond, and fatty acid containing three double bond. Now tell me, will there be change in the composition of the membrane which of the E. coli, which is shifted from 30-degree temperature to 10-degree temperature? And if there is any change in the membrane composition, what should be there? It's a very interesting question based on what you are learning today, and you should be able to answer. So when you are growing at low temperature, the bacteria is phasing, what will happen? The bacterial membrane is trying to become rigid, isn't it? When you lower the temperature from 30 to 10 degree, they are becoming rigid. Now, cell will try to condition itself in such a way that this rigidity should be avoided. So what will happen? So at lowering
Sir,
temperature,
prefer the long chain fatty acids in the structure
It
because
is a
that would increase the fluidity.
no. How
Sorry, the
long?
short chains. The short
Short.
chains would actually
The
increase
long
the
chain will
fluidity.
not increase fluidity. The short chain will increase fluidity.
Yes,
And
sir.
what it will do is that it will incorporate short chain fatty acid, and it will also incorporate the fatty acid, which is
Unsaturation.
unsaturated one. On the other hand, if I increase the temperature, say instead of 30, I make it 40 degrees temperature. So what will happen? As you said, the long chain
Then
fatty
long
acid will
chains.
get incorporated and the saturated fatty acid will be incorporated. Rest will remain in the medium. So this is a very interesting experiment one can do and can demonstrate the observation or the things which we are discussing today. Now I pose this question very differently. You know reindeer, which is in the very cold temperature, and what people they have done is scientists they have tried to analyze the fat composition of the food which is embedded in the ice and the upper part of the food. What do you expect? In the lower part of the food, what kind of fatty acid you should have? It is getting solidified, so what you should have?
Unsaturated and short chain.
Yeah, so that is what happens. So this is a very clear example where the membrane fluidity is controlled by the fatty acid. Now let us talk about the cholesterol. Now for explaining the cholesterol, let me go back to the first slide and try to explain it. Yeah, here what you find is that the cholesterol is embedded in the fatty acid. So cholesterol is not present in the bacteria, but it is present in the animal cell. So in higher organism, you will find the cholesterol which is present. So what cholesterol can do? Cholesterol, if I look at this molecule, I have shown you the structure. This cholesterol, it is interacting with the fatty acid. So what you have is you have very long chain fatty acid and a small, or a very long chain hydrophobic groups and very small OH group, which is over here. So what cholesterol does it that it interacts with the fatty acid. Suppose your fatty acid is as such that it is becoming fluid. That means you have a high temperature or you have too much of fatty acid which is making the membrane fluid. Cholesterol stops them from becoming too fluid. Why? Because there is a lot many groups which are interacting with the fatty acid, and if I increase the concentration of these cholesterol molecule, they will stop because they interact with the fatty acid. So these are non covalent hydrophobic interaction with the fatty acid, so they will stop them from becoming too fluid. And that's one role. Now I go to the another extreme where the cholesterol is present in a fatty acid chain where I am trying to put the membrane at the lower temperature. So at a lower temperature, what will happen? The membrane will solidify. And suppose you solidify to a very great extent, what should happen to the membrane? If you have some liquid material, I'm not taking the correct example, you can have a thin film. Suppose you form a thin film of a fluid, of a hydrogen, and now you take the gel, remove all the water. Have you observed this phenomenon? You take the gel, remove all the water. Have you observed this phenomenon? You take it, let for some time in such a way that its water is removed and it's become solidified. And if I go to another extreme where I remove all the water, what should happen to the gel? Have you seen a media in your plate where you have left for you forgot and you left it for longer time? What happens to that medium in a plate? Have you done? Okay, you have. Sorry, you have not done the experiment, but you might have seen somewhere. You leave the gel for a long time. What can happen to the gel? Generally, if I remove all of them, it starts cracking. So if I make the membrane too fluid, sorry, too rigid, under the rigidifying condition, the membrane starts cracking. So under those conditions, if I have the presence of cholesterol, this will help the membrane not to crack. Why? Because they are embedded in between and they will interact with the fatty acid. Fatty acid will not have very strong interaction with each other because of the presence of these bulky molecule, and that is why they will not crack. So in summary, we can say that cholesterol provides both under both the condition at a very high fluidic condition as well as in a rigidified condition. It maintains the or it controls the fluidity in such a way that under the extreme of low temperature it doesn't crack, and under the extreme of a high temperature to some extent it does not flow. Is it clear? Have you understood the concept?
Yes, sir.
Okay. So it is like, suppose I have a membrane, I make all these fatty acid to interact very strongly, they will crack. Or if I make them too fluid, all these lipids which are present here, sorry, all these proteins which are present here, they will flow from here. They will not be contained in the membrane structure. Okay, so this is about the transition temperature. Now, this is something which shows you, it's just giving you example that how this ordered and disordered structure look like. So what you have is that, and heat, how heat is giving this transition. So these are two extreme states of bilayer lipid. First one is a liquid ordered state. So this is a liquid ordered state, and this is liquid disordered state. So in first case, what you see is that polar head groups are uniformly arrayed. So this is the lower, in such a state that it is a relatively ordered state. And the acyl chain are nearly motionless and packed with a regular geometry. So you look at the geometry, depending on what is the composition, the geometry is maintained. But what happens in the liquid disordered state? It is a fluid-like state. Fluid means their motion is very fast. And what happens that fluid state, acyl chain, undergoes much thermal motion and have no regular organization. You don't see any one of them regularly organized. Some are tilting here, some are here. So that way they create a lot of space, and that is why, because of no regular organization, the state of a membrane lipid. Uh, and so the question is, what kind of a state of the membrane you have, whether L0 state or LD state. So what you find is that the state of the membrane lipid in biological membrane is maintained somewhere between these extremes. So it will somewhere here. It will neither be solid ordered state nor it will be disordered state. So at 37 degree, what you will have is you will have more or less the state which is in between. At high temperature, you will have liquid disordered state, and at very low temperature than their physiological temperature, you will have liquid ordered state. Okay, so this gives you some idea that how membranes they are present, how they look like. This part I have already discussed, and it is important that the asymmetry of the membrane should be maintained. And as I said, that for example, you have a plasma membrane on the outer side. If you have something which has to transport inside, they will be present here. The one which is transporting things outside, they will be present on the inner side of the membrane. So this example is clarified when you learn about the sodium production transport. So suppose sodium ion has to move out, so you will have a receptor for sodium ion over here, and it will be pumped out of the cell. Whereas the one which is transporting potassium ion inside, their receptor will be present on the outside. So what you have is that you have asymmetric composition of the membrane, which is outer membrane and inner membrane. Inner membrane has a binding affinity for sodium ion, and this is pumped out, whereas the outer membrane has an affinity for potassium ion, and it is pumped in. Similar way, you have asymmetry of the lipid molecule as well. Now, this is the last slide of today's lecture, and it explains that the membrane, if I look at any biological membrane, they will have certain domains. Do you remember the concept of domain in proteins? So if I look at the protein alone, you have certain domains, which becomes functional assembly within the protein. So suppose I have a full protein, you will have certain domains which will be present in a class of the protein. Now, which is assembly. Now here is a certain microdomains which are present in the membrane. And what are these microdomains known as? These microdomains are known as a raft. So this is a very relatively new concept. It was not, known earlier that the membrane lipid or membrane is also having a specific structure. Now, in this, what you find is that there are certain portion of the membrane which is enriched in sphingolipids and cholesterol. So, membrane part which is rich in sphingolipid and cholesterol, they are termed as a raft in the membrane, and these are basically the microdomain. Microdomain means a smaller area which is unique, which is not similar to the rest part of the membrane. And here what you find is that the for example, here I have shown you cholesterol presence, and they will also have these rafts. They are also having some specific proteins which are present. For example, on the outer side raft, you always have a kind of a protein which is known as a GPI link. So what is the GPI? G stands for glycosylated, P stands for phosphatidyl, I stands for inositol. So they have GPI linked protein which is present on the outer surface. Whereas on the inner surface, there is another fatty acid linked protein which is present. So here, what you have? You have a protein which is going inside and coming outside on the inner surface. It is not connected with the outer surface of the membrane. It has acyl groups. So when I talked in the first slide that there are certain fatty acids which are linking the protein by covalent interaction. So these are the examples. Say prenylated protein, they are interacting with the membrane. It is a covalently bound. This caveolin, this is again bound by acyl group like palmitoyl group or myristoyl group. So what you have is that, or here doubly acylated protein. So two acylated groups are binding. So this way, the membrane protein are attached with the membrane by covalent interaction. So I have taken one example here, another example of caveolin. So you will find that outer leaflet will have a different domain than inner leaflet. So inner leaflet is enriched with caveolin, whereas outer leaflet is enriched with the GPI linked proteins, and they form a specifically enriched domain of the membrane, which is made up of sphingolipid and cholesterol. So with this, I'm going to stop. Okay, one more thing over here is that suppose something is not attached with the area, but still it is a protein attached with the membrane and does not have any specific enrichment of this sphingolipid or cholesterol, we do not consider them under the raft. So they will be excluded from the raft. So any protein which is not linked with the membrane and has no high concentration of sphingolipid and cholesterol, they will not be included under raft. So raft is a specific microdomain stabilized using association of sphingolipid and cholesterol in different leaflet, could be outer, could be inner, and they are specific in terms of their protein. So I'm stopping at this point. You can give, if you have any question, I can address.
So for the last one, the
Yeah.
last part that you explained,
Hmm.
so isn't the protein attached to the membrane?
It is attached to the membrane, but what you have is that this area. So here, if I analyze the lipid, you will find that they are not rich in cholesterol. So why they have, yes, this is which is required for the raft. But other than that, you have to have a cholesterol and you have to have some of the sphingolipid, the green one, for example, here, should be present to make them as a microdomain. So two conditions. Number one, that it should have high concentration of sphingolipid and cholesterol, and and the protein may or may not be attached here. So protein attachment is found in such area. And that is why they have distinguished that, for example, in outer raft, you find GPI linked, inner raft, you find caveolin. But for example, if something is, for example, in this area, if you analyze and you find that sphingolipid and cholesterol is not rich, we don't consider in the microdomain. Same way here. So microdomain has a important thing that it has a enriched with sphingolipid and cholesterol. Okay. Any
Yes,
question?
sir.
Any other query? So if there is no query, in the next class, I will talk about the functionality of the membrane, and we will talk about the function, and we'll continue with this membrane.
So I
So
have a question.
membrane structure function is over. Now structure part is over, function part will start. Yeah. Any
So
question?
uh, what is the syllabus for the
See,
minor?
whatever is covered for the before minor, that will come in a minor.
Okay. Thank you.
Okay, then I will, sign off, and thank you very much.
Thank you, sir.